Well, welcome back, Biology 1406. During this presentation, we're going to be covering Chapter 13, which is the molecular basis for inheritance, kind of getting us into understanding how DNA works and how genetics work a little bit more on the molecular level. A lot of people that also need to be recognized as well. And this is probably the most infamous picture of Watson and Crick with their DNA molecule showing what they think DNA looks like. So in the early 20th century, we were identifying a lot of molecules, and that was a major challenge for biologists to understand. We understood what molecules were, but we couldn't quite visualize what it was that they looked like. So there was an individual named T.H. Morgan, and his group showed that genes were located on chromosomes. And we also knew that there were two components of chromosomes, DNA and protein. And so those two things became candidates for genetic material. So we didn't know yet that DNA was genetic material. Until the 1940s, the case for proteins seemed stronger, but we know today that proteins are not what contain our genetic material. It's DNA that does that. So the role of DNA in heredity was worked out by studying bacteria and viruses that infect them. And you guys probably remember this back from exam 2, maybe, chapter 17, when we covered bacteriophages. The discovery of the genetic role of DNA began in 1928. And he worked on 2 strains of bacteria, and he was looking at one that was pathogenic, so a bacteria that was harmful, and then one that was harmless. And so he mixed heat-killed remains of the bad bacteria, the pathogenic bacteria, into living cells of the harmless strain. And what resulted from that was that some of the cells, the living cells of the harmless strain, became pathogenic because they now contained the heat-killed, the dead pathogenic strains. And so he called this transformation. It was a phenomenon, and he was able to now define as a change in genetic and phenotype due to assimilation of foreign DNA. So we can change our genotype and our phenotype, genotype being the genes, phenotype being the way that we look, and that is due from combining foreign DNA into known DNA. So this is just an image of that experiment that he did, where he gave pathogenic cells to a mouse. The mouse gets sick and it dies. He gives non-pathogenic cells to a mouse. That bacteria doesn't kill the mouse. He gives heat-killed non-pathogenic strains to the mouse. The mouse doesn't die. That's what we would understand it to be. And he did that so that he could show that heat is not what the heat introduced into the strains is not what is hurting the mouse. But when you killed the pathogenic strains with heat and combined it with the non-pathogenic strains, the mouse still died. So he introduced pathogenic genes into the non-pathogenic genes. There was also an individual that you may or may not know the name of, Oswald Avery, and he was known for identifying transforming substances as DNA. Another individual named Oswald Avery and his group identified the transforming substance as DNA. So the thing that changed that non-pathogenic bacteria to being bad, he'd said that's DNA. Many biologists still remain pretty skeptical, and this was mainly because we didn't know a lot about what DNA was at that time. So more evidence for DNA as genetic material came from studying viruses that infect bacteria. And you remember bacteriophages or phages are widely used in molecular genetic research, mainly because it's very simple. It's very easy to study. A virus is DNA or RNA. Remember, it's enclosed by a protective coat. And those viruses can then take over the cells. They can be injected into the cells. So if you remember from chapter 17, the look of that bacteriophage, right? It lands, it docks itself down, and it injects its genetic material into our cells and then or into other cells. And then the virus enters the body or enters the cells of the body, and something happens. You become ill, you become sick. There is a reaction that occurs because our cells are trying to get rid of that foreign DNA. So in 1952, Alfred Hershey and Martha Chase started showing some evidence that viral DNA can program cells, that they can make cells, once they get into the cells, they can make cells do something, they can hijack them. And they designed this experiment and showed that only the DNA of a T2 phage and not the protein of that phage enters E. coli. So what they discovered was that it's DNA, it's not protein, that is responsible for genetic material or genes, provides genetic information. In 1950, a guy named Erwin Chargaff had reported that DNA composition varied from one species to the next, and so that was just additional evidence that DNA is the genetic material and not that proteins are. So you notice that there's a lot of around the same, decade of time, around the same period of time, there's a lot of people doing different portions of the same research. And so by being able to take pieces of everybody's research, you can come up with a theory, or you can come up with outstanding evidence to prove something is correct versus the way that people had always thought that genes were part of protein. And all of this information helped us discover that it's actually DNA. The Chargaff's rules, his two findings became known as Chargaff's rules, and that was that base composition of DNA varies between species. So our DNA is different than the DNA of dogs and the DNA of insects. We're different. Our DNA is what makes us different. And that in any species, the percentage of A and T bases are equal to C and G bases. So we haven't talked about this just yet, but all the pieces, so A and T and G and C are parts of DNA. That makes no sense right now, but it will a little bit later on. So he was important for discovering that the percentages are equal. So this is what we're talking about when we talk about A and G and C and T. So the structure of DNA is that think of it as a ladder. And so the sides of the ladder that you hold to when you climb up the ladder is a backbone. And it's a phosphate backbone made up of deoxyribose and a phosphate group that connects each ribose to one another. In between the rungs of the ladder are these nitrogenous bases, and they are either thymine, guanine, cytosine, or adenine. So that's where the A, T, G, C come from. And they will bind with another nitrogenous base, which will then be attached to the other side of the backbone. So what you're looking at right now is if someone had a ladder and they buzzed it right down the middle and you only have one part of it. To make the full ladder, the full structure of DNA, you have two backbones and two nitrogenous bases that are linked to one another. And so what Chargaff found was that you have equal percentages, A and T with G and C. Now we go to Watson and Crick. And so Watson and Crick were the first to determine the actual structure of DNA. And they used this based on everyone else's information. And specifically, they used it using the information from Maurice Wilkins and Rosalind Franklin. They were using a special technology called X-ray crystallography, and it studied molecular structure. And so Rosalind Franklin took a picture of a DNA molecule using that technique. And she has posthumously been recognized as the actual person to discover the way DNA looked. But it took Watson and Crick to look at that picture and determine that, oh, this is a double helix. I mean, that's kind of mind-blowing if you think about how clever and intelligent that they were.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
Running means. But if they hadn't built that pattern, then they wouldn't have been able to determine that. So at first, Watson and Crick thought that the base paired like with like. They thought A and A, T and T, G and C, and so on. But what they found out was that that is not provide the uniform width, because some of A, T, G, and C are different sizes. So some are, a couple of them are smaller and a couple of them are larger. And so really what you had to pair was big with small, small with big, and so on. You couldn't go small, small, and big, big. You would have this kind of warped backbone, and the ladder would not work properly. So what they found was that, so one is a purine and one is a pyrimidine. This is the way that I remember it. Purine is a smaller word, which means it's a bigger molecule. Pyrimidine is a bigger word, but it's a smaller molecule, so it's opposite. And so purines and pyrimidines pair up, and purines will not. Pyrimidines and pyrimidines will not. So the purines are bigger, and if they pair up, then it's too wide. It doesn't fit into the double helix. If pyrimidines and pyrimidines pair up, it's too narrow. They cannot pair up. So what he found was that it fits perfectly. What they found was that it fits perfectly if purine and pyrimidines match up with one another. That fits within the backbone perfectly. And what he found was that adenine and thymine, so AT, pair together, and GC pair together. And I remember that because AT spells at, and G and C are both curved, and so they just seem like they should match one another. A and T are pointy, so they pair up. G and C are curvy, and so they're going to pair up with one another. And that also corresponded and supported what Chargaff found, which is that if you have X amount of A's, you're going to have the same amount of T's, because they are paired up with one another, so it should be equal. And if you have X amount of G's, you're going to have the same amount of C's. So just to kind of recap, pyrimidines match up with purines. Our pyrimidines are thymine and cytosine, and our purines are guanine and adenine. Adenine always matches with thymine, guanine always matches with cytosine. So I would kind of write that down and make sure you remember it. Purines are the larger molecules. Adenine, guanine. Pyrimidines are the smaller sized molecules, thymine and cytosine. A and T always shake hands, and G and C always shake hands. And they're held together by hydrogen bonds. So when we talked about chemical bondings in the past, we said that hydrogen bonds are relatively weak bonds. But because a DNA strand is so incredibly long and you have all of these hydrogen bonds, it becomes incredibly, incredibly strong. So remember, we talked about how it was not really decided, is it proteins or is it DNA that makes up your genetic material? Well, we did find that proteins are part of chromosomes and they do play an important role in replicating DNA and repairing DNA. So DNA will replicate, we know it does, because our cells have to replicate. And it also has to repair itself at times too. So this is kind of a very basic model for DNA replication. It is known to be semi-conservative, which means that DNA will split down the middle, so the purines and the pyrimidines will separate, leaving behind one base pair with a backbone on one side, one base pair with a backbone on the other side. And then those two separate pieces, those two halves, will then replicate. So it's semi-conservative because a new DNA strand has a new part but an old part. So they conserve half of themselves. So they separate and then repair or rebuild. Since two strands of DNA are complementary, each strand stores the information necessary to construct the other. In DNA replication, the parent molecule will unwind and the two new daughter strands are built based on the base pairing rules, with A and T, G and C pairing up. So Watson and Crick's semi-conservative model, semi-conservative because half of it's used, reused again, of replication predicts that when a double helix replicates, each daughter molecule will have one old strand and one new strand. Competing models were the conservative model where the two parent strands rejoin, and then there was another model that said it was a mix of the old and new, but it has since been discovered that Watson and Crick's semi-conservative model is the accurate model. So there was still a lot of work going on trying to determine exactly how DNA replicated. And other experiments supported that semi-conservative model. The copying of DNA is remarkable in its speed and accuracy. There's more than a dozen enzymes and other proteins that participate in it. We're going to talk about what some of those are. Much more is known about how this replication machine works in bacteria than in eukaryotes, and most of the process is considered to be similar between prokaryotes and eukaryotes. So by studying something that's easier to study, we can understand how it works in a body like ours. So replication will begin at a spot that's called the origin of replication. And basically the two DNA strands will separate and it makes a little bubble. And then at the end of each bubble is a replication fork, a Y-shaped region where it's kind of separated and the DNA is unwound. For the long DNA molecules in eukaryotes, we'll have multiple bubbles that form, and so this speeds up the replication process.
DNA replication, an overview. In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules. Here's a simplified close-up of replication, showing what happens along one of the template DNA strands. Using a supply of free nucleotides, DNA polymerase assembles a new DNA strand along the template strand, following the base pairing rules. Adenine pairs with thymine, and cytosine pairs with guanine. Now let's look at the DNA replication process. Replication of a DNA molecule begins at a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we've untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding 2 identical daughter DNA molecules. Now, let's review the synthesis of DNA by assembling a new DNA strand. We'll use the top DNA strand as the template and choose from the free nucleotides below. Note that D stands for deoxyribose and R stands for ribose. We're only using the deoxyribose nucleotides because we're building DNA, deoxyribonucleic acid.
So remember I mentioned that there's lots of proteins and enzymes that help participate in replicating DNA. So helicases are enzymes, and remember enzymes are proteins that help reactions occur quicker. So a helicase is an enzyme that untwists the double helix at the replication fork. Then single-stranded binding proteins will bind and stabilize the single strand. Topoisomerase relieves the strand caused by tight twisting ahead of the replication fork by breaking, swiveling, and rejoining the DNA strands. So helicase untwists, single-strand binding proteins stabilize, and topoisomerase helps rejoin and put it all back together again. So there you can see, if we follow it along, so topoisomerase breaks, swivels, and rejoins the parent DNA ahead of the replication fork, causes it to unwind. Then the helicase unwinds the bubble right there, forms that bubble. We have that Y-shaped replication fork. Primase synthesizes RNA primers using the parental DNA as a template. So, and then single-stranded binding proteins stabilize the unwound parental strand. So lots of proteins are involved. It's a very complicated process. Bubble forms, replication forms, but beyond where the bubble is, you have to have that topoisomerase to help rejoin and stabilize, because it's in a form, by having untwisted, it's in a form that it doesn't want to be in. It's not in its most stable position. Enzymes that synthesize DNA cannot initiate synthesis of a polynucleotide. They can only add nucleotides to an already existing chain. And the initial nucleotide strand is a short RNA primer. Primase starts an RNA chain with a single RNA nucleotide and adds RNA nucleotides one at a time using the parental DNA as a template. Enzymes that are called DNA polymerases catalyze, make things happen, right? The elongation of the new DNA at the replication fork. They add nucleotides to the 3' end of the existing chain. Most DNA polymerases require a primer and a DNA template strand. Each nucleotide that's added to the growing DNA consists of a sugar attached to a base and 3 phosphate groups. DATP is used to make DNA. It's similar to ATP, but it's for DNA, so that's easy to remember because there's a D in it. But it is the energy to make all of this happen. So there you go. There's kind of the making of a new DNA strand. It has split, and so we have our parent molecule here, then the darker blue. We have a new strand being built from the 5' end down, and it's DATP that's helping to make that occur by providing the energy for it. So we say that DNA replicates in an anti-parallel elongation, and that means that it So because DNA polymerases add nucleotides only to the free 3' end of the growing strand, the strand can elongate only in the 5 to 3' direction. So it goes from 5 to 3' down. Along one template strand of DNA, the DNA polymerase synthesizes a leading strand continuously, moving toward the replication fork. Only one primer is required to synthesize that leading strand. So let's watch an animation showing you how that occurs.
Let's now look at the process of DNA replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The two strands would naturally tend to rewind, but are held apart by molecules of a single-strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction. The strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary DNA nucleotides to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA. Next, the enzyme DNA ligase links the Okazaki fragments to form the lagging strand. Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication. Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
What's amazing is that errors in completed DNA molecules are only about 1 in 10 billion. So this is an extremely precise and well-oiled machine because mistakes simply just don't happen very often at all. DNA polymerases proofread newly made DNA and replace the incorrect nucleotides. And so that's one of the reasons why mistakes don't actually make it to production, in a sense. In mismatch repair of DNA, other enzymes correct errors in base pairing. So there's multiple factors that are helping proofread that the DNA was made correctly. A hereditary defect in one such enzyme is associated with a form of colon cancer. So there are genetic components to some types of diseases, including certain types of cancer as well. And it's because that DNA constantly makes itself incorrectly. DNA can be damaged by exposure to harmful chemical or physical agents such as X-rays. You know that you don't want, if you're pregnant or if you're thinking about becoming pregnant, you certainly don't want to ever expose yourselves to X-rays. And so that's why you wear, if you're an X-ray technician, that's why you wear the big lead apron so that you're protecting the areas where DNA replication is occurring very often in the production of gametes. DNA bases can also undergo spontaneous changes. And in many cases, a nuclease cuts out and replaces DNA stretches. And in many cases, a nuclease cuts out and replaces damaged stretches of DNA. One such DNA repair system is called nucleotide excision repair. So DNA repair enzymes in our skin repair genetic damage caused by ultraviolet light from the sunlight. But because you're constantly replicating that DNA and fixing things, that's when things can go wrong, and that's how skin cancer occurs. So once again, the error rate after they're doing all this proofreading is extremely low, but it's certainly not zero. So sequence changes may become permanent and can be passed on to the next generation. These changes or mutations are the source of genetic variation upon which natural selection may operate also. So at the end of nucleotide sequences, the ends are called telomeres, and they kind of are shown here, right, at the kind of brighter colors. Telomeres don't contain any genes, but they do consist of multiple repetitions of one short nucleotide sequence. So they don't prevent the shortening of DNA molecules, but they do postpone it. And it's been proposed that the shortening of telomeres is connected to aging. If chromosomes of germ cells become shorter in every cell cycle, essential genes will eventually be missing from the gametes that they produce. An enzyme called telomerase catalyzes the lengthening of telomeres in germ cells. Telomerase is not active in most human somatic cells, though, but it does show inappropriate activity in some cancer cells. And so we're studying it as a target for cancer therapies by allowing that telomerase, by the telomerase allowing the cancer cells to continue to allow replication is not a good thing, right? So if we can target them, and if that in some way stops cancer cells from replicating, then that can help reduce tumors or reduce the spread of cancers. The bacteria, so in concept 13.3, we're looking at that chromosomes consist of a DNA molecule packed together with proteins. When you look at bacterial chromosomes, they're double-stranded, circular DNA molecules that are associated with a very small amount of proteins. But eukaryotic chromosomes are linear DNA molecules, and they have packed full of tons of proteins. In bacteria, the DNA is supercoiled, and it's found in that nucleoid region. In eukaryotes, of course, it's found in the nucleus. Chromatin is a complex of DNA and protein, and so it's found in the nucleus of the eukaryotic cells. Remember, before chromosomes become condensed during mitosis and meiosis, they're just chromatin. They're just kind of a jumble, bunch of spaghetti, just kind of really messy. The first level of packing that DNA into packing the chromatin, making it become more condensed, involves 4 main types of proteins that are called histones. So the nucleosome is DNA that's wound around 8 histones, so doubles of the main histone types. And there you can kind of see, you can kind of watch the progression of how it's just kind of real loosey-goosey, and then it starts to become more condensed and starts to get wrapped and wrapped and wrapped, and now it looks like the 2 chromosomes that we recognize that are made up of all of that genetic material.
Several levels of DNA packing make up the compact chromosome seen in a metaphase cell. Even when a cell is not dividing, some of the DNA is folded tightly. The degree of DNA packing makes certain genes available or unavailable for transcription. Chromosomes consist of loops and supercoils, which are made up of tight helical fibers. These fibers are composed of nucleosomes, which consists of DNA wrapped around protein molecules. In the fiber, the DNA is unavailable for transcription. Here we can observe the unpacking of DNA, which makes it accessible for transcription. Each nucleosome consists of a DNA segment wound around a protein core of 8 histone molecules. Histones are involved in switching genes on and off. Chemical modifications of DNA and histones may affect chromatin structure and access of RNA polymerase to specific genes.
DNA double helices are shown in purple. Histone core proteins are shown in green. Credit: Jeff Harden, University of Wisconsin-Madison. Rendering created using Pymol. http://www.pymol.org.
At interphase, during mitosis, most of the chromatin is compacted, and then it continues to be folded a little bit further. So we saw that in this image over here. Even during interphase, the centromeres and some other parts of the chromosome are pretty highly condensed, similar to metaphase chromosomes. So the condensed chromatin is called heterochromatin, and it's the more dispersed and less compacted chromatin is called euchromatin. Dense packing of the heterochromatin makes it largely inaccessible to the machinery responsible for transcribing it. Chromosomes are dynamic in structure, and a condensed region may be condensed, it can be loosened, it can be modified, and it can be remodeled for various cell processes. And histones can undergo chemical modifications as well, and that results in changes in chromatin organization.